welcome Sheng-hong. Okay, Dr. Chen. Okay, 谢谢。你要用这个还是要用这个？用这个。谢谢。
So thank you, Jiafu, for your very kind introduction, and everyone for coming here. So I think there's a reason. It's actually not courage. It's actually just naive. But I always remember, that when I was doing my postdoc at UCSF with Keith Yamamoto, and there was a graduate student. So I was talking to him. Oh, I want to do this project. This is relatively conservative project, and I can get a publication out. We were chatting, and then he was like, "Why are you doing that?" And I was like, I was having some strategic thinking about this. And he was like, "No, you should never do that. You should do whatever you love every single second, if possible." And then that moment, just like very inspiring for me. And then, so I asked myself, when I become a PI, no one is asking me to get anything done. I should really pursue what I want to do. But another side of this fact is, I actually didn't know what I want to do. But what I want to do is actually to do something different. I want to do something no one is trying to do at that moment. I don't know where it's going to lead us. But I have, probably because of my previous mentor's encouragement and training, make me feel that I probably can get something done. But I want to do something different. And I actually think that is something I really appreciate in giving a Sinica, or actually research in Taiwan, is that we are not in a research hub that, I don't know if you guys know that, in Boston, in biomedical research at least, when the discovery of CRISPR, for example, occurred, everyone talks about CRISPR. And you are more like, you cannot immune from CRISPR because it's everywhere. But in Taiwan, I feel we don't have this kind of mainstream that is driving or pushing us, so that I actually really appreciate and enjoy the moments that we can stay in our labs thinking about some questions to the very fundamental level, that for us to do something that we can enjoy and potentially can be important. But I think we can have fun during the process. So I think, just to follow what Jeff was saying, it's not that always romantic, I guess. But some, in retrospect, yeah. So my lab is called Labs of Cell Dynamics. Actually, I feel that I probably don't need to elaborate too much on this, because these are all physicists here. But I do want to say that dynamics is really important in biology. We actually leverage a lot of tools and theories that has been developed by theoreticians and physicists to understand biological systems. But I still want to talk about why it is important, because a lot of time in biology, we have snapshots like this, and that's our understanding of a so-called fact. But the truth is that if we have dynamics or time axis, then we have very different understanding of the events, right? So that happens to biology all the time. If you go to the thesis defense or some kind of progress report in biological field, and the student would say, I cannot repeat this result. And it's so noisy, because maybe I collect samples sometime in the morning at 10, sometimes afternoon at 2, and as a result, it's very different in terms of the dynamic process we can capture, right? So, but we are especially interested in the dynamics between metabolism and signaling events. And in biology, signaling event a lot of times relate with some proteins like kinases, the enzymes that can put some phosphate on other proteins. That's like conventional signaling events, or transcription factors, those specific kind of enzymes that can turn on the expression of hundreds or thousands of genes. On the other hand, metabolism is more about how do we break down some nutrient from the environment and then make them into energy or useful resources for the cell. And these two fields in the past, I think signaling especially have been very so-called dominant in the past 20 years or so because of the development of molecular biology, allow us to have a lot of tools to actually control the signaling events. On the other hand, people always think that metabolism has been established 20 years ago because so many Nobel Prizes has been given to people study metabolism. So it's already fixed. But the truth is that it's not true. Metabolism is actually also very dynamic. And these two kind of events are interacting at very different time scale a lot of times, and then can result in very interesting phenomena that we are just about to discover, I think, in the near future. So my lab is interested in dynamics that across two dimensions. That is, one is time that we are interested in knowing. Some of the molecules in the cell that can undergo very interesting changes over in terms of the level over time. In this case, this will be a so-called a system called oscillators that will keep going on and off when cells are exposed to stresses. It's very interesting because a system like this can be so robust. For example, this specific molecule called p53 can oscillate in individual cells for months. Yes, so there is a reason why the system evolved so that it can oscillate so robustly. Of course, it's not robust in a quantitative way that it always maintains the same amplitude, but the period relatively is very, very robust. So there are a lot of information that biology is using those dynamics to actually kind of express certain functions in the cell. Okay, we are interested in knowing this, and now we have a lot of tools that we can monitor those signaling dynamics in individual cells and knowing what the cells are making decisions. And a lot of time we can find those dynamic behavior of the molecules are actually encoding some information for cells to make decisions. Okay, so cells are making decisions through this dynamics of the signaling molecules. It's not only about changes over time, but cells can also have a system like switches or so-called bistable systems, like a multi-stable system. It can be like a toggle switch, can turn on or turn off, and it has some memory and has some hysteresis properties, all that. So those are very interesting phenomena in biology that actually is very fundamental and important. For example, a fertilized egg now can develop over a course of years and become so many different kind of cell types by turning on specific transcriptional programs and through a series of switches like this. Okay, so I think those are interesting phenomena that we want to understand better. In terms of space, we can also see sometimes cells are behaving very interestingly and mysteriously like this, that they can, without any superimposed guidance, they will form this kind of tornado-like structure, right? So, in fact, this is a phenomenon that firstly proposed by physicists. They draw an analogy of this kind of behavior as pneumatics or liquid crystals. They think that cells can behave like liquid crystals, that in a two-dimensional space or three-dimensional space, they can form certain topological patterns that may be of biological importance. So in this regard, we actually are interested in knowing this kind of behavior. What can be the biological significance or function behind this kind of behavior? And finally, I think we are also interested in how cell can actually spread the signal over long distances through a mechanism called trigger waves. Basically, it's a domino effect of how cells turn on a specific biochemical reaction, and that chemical reaction then leads to the so-called domino effect, like turning on of this chemical reaction over space across cells. Okay, and this will be a major topic I'm going to cover today. So today, before I start, I would like to really thank the people who really did the work. So Hannah Katrinako, she just graduated from the TIGP MCB program, and she is the person who did most of the experiments that I'm going to show you today. Jiazhou is a postdoctoral fellow. He developed the mathematical models as well as a lot of image analysis for us to understand this dynamic process of trigger waves. And finally, we also did use a lot of chicken embryos for these experiments. So a fundamental question we are interested in knowing is what's the relationship between structure and function, right? So if we look into this sculpture, you will feel that this person using every single fiber of this muscle to contribute to the thinking process, right? The truth is that the function is a lot of time related with the structure. And this has been actually observed a lot, like in the mid-19th century by scientists. They just get the insect embryos and look into the microscope. And they could see that during the development, you need, from one cell, you need to become hundreds, thousands, millions of cells. So active cell proliferation and division, that's for sure. But they also found that a lot of the time the cell would die. You see a lot of cell deaths as well, okay? And maybe the cell death is important to form certain kind of shapes, for example, a tunnel or a tubule shapes, and you need to somehow carve all those tissues out, right? And those kind of observation actually, these inspire people to find what can be the molecular basis of this cell death during development. And so a leading pioneer in understanding the cell death during development is Robert Horvitz. He used C. elegans to understand what are the pathways, so-called molecular systems that actually contribute to the cell death during development. And he could find, so those arrows are the cells that undergo cell death, okay? And C. elegans is the little tiny worms that you can use genetic approach to actually identify what can be the genes that are important for the cell death here. And he found this kind of cell death called apoptosis. It's a so-called programmed cell death. And because of this, he got Nobel Prize for it. And then, but if you pay attention to this, you will see that a lot of those apoptotic-induced cell deaths here are usually at the end of the lineage. So maybe I can give a brief introduction of this. So P12 is like initial progenitor cell, then it divides, and then this cell will divide again, and one of them will die. So he can use this kind of ways to track, how the cell would die in different kind of lineage process, and also find what's the molecular pathways. But that was around the mid-20th century, and around the same time, there's another scientist called John Souders. He didn't use C. elegans, which is a very simple worm. So worm is special in a way that there is a fixed number of cells in the adult C. elegans. Every single C. elegans has the same number of cells. So everything is pre-made and very nicely programmed, but which is very different from complicated organisms like vertebrates. Okay, so John Souders used chicken embryo as a system. And what he found is that he say, during the development of the chicken embryo, there can be very abundant cell deaths, and that can eliminate organs and tissues that are only temporarily useful during embryonic life. When we talk about organs and tissue, that's very likely millions of cells. You have a lot of cell deaths happening in the complex organisms like chicken embryo. Okay, so but large-scale cells is also not only happening during development, like for brain trauma or like heart diseases, kidney failure. A lot of times, especially really with aging diseases, you see large-scale cell deaths occur, right? So then the question we try to raise is, how does this large-scale cell death occur? And what can be the mechanism underlying large-scale cell deaths? Okay, we actually we get some initial clue actually from physicists. So Max Delbrück is one of the physicists who proposed, firstly, that the enzyme system can have alternative steady state. In other words, given the same physical and chemical status, enzyme 3 can actually occupy a state of 1 or 2, and they are both stable. That's why it's called bistable. Okay, it was kind of an intuition from Max Delbrück, but then later on, through a series of both experiments, Aronovik's molecular biologist has tried to do experiments to prove this multi-stability occurs, and later on, actually, a lot of theoretical physicists trying to use mathematical tools to understand the occurrence of multi-stability. Actually, after more than 50 years of efforts, Alex van Oudenaarde, he at that time was in MIT, he brings theory and experiments together. He firstly showed that in E. coli, there is a system called leg operon. This system can be multi-stable. It can be turned on or turned off. It's so-called all or none response, and it has memory, has hysteresis, all these kind of properties. By doing experiments in single E. coli cells, as well as doing theory, trying to understand what's the phase diagram of the system, how can you turn the system to be monostable or not. But basically, for biologists at least, it means that the E. coli has the memory of what's the nutrient in the environment. If they are exposed to certain nutrient for frequent enough, they will remember the nutrient is like a frequent nutrient in the environment, so they will turn on the leg operon to digest the nutrient, so-called lactose. But if not, then they will remain low. So it's like a switch. Yes. Quantum tunneling. Yes, and your question is?
I mean, you have
Stable states. Mm-hmm.
and it's well known in
Uh-huh.
that's why of this
I see. Well, that's cool. I'm going to talk about,
thermal fluctuation.
I see. Yes, right. So I'm going to talk about, we found that the occurrence of this multi-stable, and we can actually turn it on through some kind of experimental approach later on. Yeah, right, okay, good. Right, so with that background, then we found something interesting that is also like a switch. So when we take out the glucose in the environment, what happens is the cell will start to have this kind of switch-like change in that's fine. I think you can also, by looking to these time-lapse images, you'll see this signal turning on, right? And then some cell will never turn on. And those signals actually represent the hydrogen peroxide level in the cell. So when you are lacking glucose in the environment, what happens is that the so-called redox system, which is about to control the ROS level in the cell, suddenly become a switch. And once it bypasses the threshold, it will turn it on. Because the ROS level is so high in the cell, over a certain period of time, it will burn the cell to become a bomb, and the cell will just die. Okay, so that's what we learned from this approach, that if you take out some nutrient, in this case glucose, then cell will become a switch-like system that can, once it turns on, it can generate a lot of ROS in the cell. Okay, so if I were to use an analogy, a bomb will be something like a firecracker during the New Year, right? That kids play. And then usually we will actually bring them together so that you can have a physical coupling of these firecrackers. And it can actually be pretty powerful. This is one real event. This is people, you know. When they put all the firecrackers together in space and they are physically coupled, then you can generate a crazy response, like a biochemical reaction, right? So the same thing if the cells are like a little firecracker, and when they are physically coupled, what happens? That's what Hannah did the experiment. So she tried to deprive cells with another nutrient called cysteine. And we know if you take out this amino acid cysteine then in the environment, then the cell would also become a bomb again. And then, so before the start of this movie, she actually took out the cysteine and started to image the cell. These are individual dots, like triangle shape or irregular shape. Those are individual cells. When the cell dies, they will round up like a ball, okay? And you can focus on this left side where the cell death will initiate. You see the cell die and then start to spread across space, okay? And another way to show it is to use another dye that after the cell break, then the dye will go into the cell, then the nuclear dye will actually light up. It's the same movie but different channel. You'll see the cell will die and start to propagate in space. Okay. And we were using this is a specific cell line called retinal pigment epithelial cell. This is at the back of our retina. But we actually try more than 20 different types of human cells. They all can have this kind of spread-like behavior. Okay, so then our hypothesis, or at least one potential hypothesis, that this kind of large-scale cell death may occur through this a specific kind of cell that's called ferroptopsis. And then the mechanism underlying this propagation is called trigger waves. So I'm going to try to explain this to you in the following several slides. What is ferroptopsis? It was actually identified in 2012, a relatively new kind of form of cell death. I was talking about cysteine, right? So cysteine, why is it so important to dampen ROS? Is because cysteine actually, after uptaking by the cells, it actually is important for the generation of a metabolite called glutathione. And glutathione is a very good, strong reducing power in the cell. It will suppress ROS in a cell, reactive oxygen species. And reactive oxygen species, there are multiple different species. They can interconvert into each other. For example, hydrogen peroxide can be converted into hydroxy radicals through a chemical reaction called Fenton reaction. It's catalyzed by iron 2 plus in a cell. And this hydroxy radical can then feed into the oxidation of lipids. And this kind of lipids usually is so-called unsaturated fatty acid. It has double bonds that can be oxidized. And once it's oxidized, then it can break. And then, honestly, right now, people still don't know. But the speculation is then it will make the membrane to be having holes. And then as a result, the cell membrane will break. And then the cell would die because the membrane integrity is broken. So, in other words, ferroptopsis depends on the iron that catalyzes lipid peroxidation. So this is a type of cell death that we call ferroptopsis. Second, trigger waves. What is trigger wave? It's quite different from diffusion, which is the signal intensity and the spread of the speed will dampen very quickly over time and space at the scale of, say, 10 cells. If it is bigger, like, say, hundreds of micrometer, then diffusion becomes very limited. Trigger wave, on the other hand, it will maintain its own signal intensity and as well as speed. So in nature, one type of trigger wave is actually just like wildfire, right? You can have, as long as you have an initial initiation of the heat, the fire, then you start to burn, and then you need to have some kind of coupling mechanism. It can be diffusion. In the case of ferroptopsis trigger wave, can mediate through cell-cell communication. Then the fire can spread to another tree in this case, and then you need fuel. The neighboring cells or neighboring trees, they need to be dry and they can be burned. So in a way, it's also behave like a switch. Once you overcome the threshold, then the tree will get burned, and then as a result, the fire can be propagating indefinitely. So we try to use this analogy. Yes, Qing. What wave? Sortical. I don't know this. But where is this? What drives the wave? Yes. 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 I think it's probably the same. The nonlinear turns comes in the switch. You really need a nonlinear response in the system so that the system can have multi-stability. Yeah, that's right. Okay, good. So regarding to the initiation, because initially when we study it, the cell would die in various different places and you have multiple initiations. So Hannah developed a way that she can use a blue light to expose to a specific subpopulation of cells. And before the start of the experiment, she would starve cells with cystine, so that we think that maybe in that case, then cells would all become a switch. They are already starved. The ROS can be turned on. And what this light is doing is to overcome the threshold of this ROS switch. So to turn on the light of those cells. And once she turns it on, what happens, they will start to spread in space. And this is a really big movie. This is 1 millimeter, right? So it's like 6 millimeter long square. And what you see already, this is like millions of cells already die. And they follow this kind of outline with a constant speed, okay? With this way, then we can know this is really not diffusion, because diffusion would really slow down at this time, this scale. Yes. Bistability does not. It is true that bistability a lot of time referred to in reversible system, but by stable system, it can also be turned on depending on the bistable regime, right? So biologically, it is right, because once they turn on, they will burn, they will die, so it's impossible to reverse it. That is true. Yes. So we did measurement of the speed. The speed is about like 5.48 micrometer per minute. In other words, about 2 to 4 minutes, the wave will go over a cell. And it's very different from diffusion of proteins or small ions. It's maintained a constant speed. I was talking about ferroptosis a way that final output is the lipid peroxidations. We can also visualize the peroxidation of lipids by a dye called CLN-BODB, and which will be represented in color yellow. So you will see, again, once we trigger it, you will see a wave of lipid peroxidation preceding the cell death occurring here with cyan color. In other words, the cell would actually have really high level of lipid peroxidation, and then they die, and probably for some reason the coupling mechanism make the cell to actually, the neighboring cell to turn on again and keep going, right? So we want to know what can be the coupling mechanism, right? So the first thing we want to know is that, does it require cell-cell communication? Yes. Sure. How do we control, the question is, how do we control the threshold or the cell density? Oh, how do we control the cell density? Yes, that's actually a really good question, because the cell density here is very homogeneous. Maybe a on-the-table story about this is Hannah, it took her more than half a year to develop this assay. By knowing how to play the cell in a very homogeneous way and how to change the culture media, I think she tried more than 100 different combinations. And eventually we can have a very homogeneous wave like this. So, but we publish all these protocols. It just takes a lot of trial and error, to be honest, to really see the isotropic spread of the cell death. Yeah. Yes. Yes. So I'm going to talk about, at the end of the talk, I will talk about some of our preliminary data where cells are self-organizing into different cell density and patterning and see how this wave will actually propagate in that context. But in this case, yes, that's a good point to mention, is that it's very homogeneous in density. All the cells are more or less homogeneous. Yeah. So I was talking about the question of what can be the coupling mechanism. Can it be diffusion, or the cells need to the cells, when they are about to die, they kind of spread something in the environment and through cell-cell communication. So we did an experiment where we tried to create physical gaps in a cell population, and we introduce cell death wave from one side and then see if it will propagate and can it overcome the physical barrier. And when the barrier is like around 100 micrometer, then it can actually go through. However, when the gap is big enough, around 150 or so, then you start to hold the wave. Okay, we do this statistic on this. It's around like 150 to 200 micrometer, then the wave will start to stop. Okay. So it means that you don't need direct cell-cell contact for the signal to propagate. And it can be some kind of a local diffusion of the molecules when the cells are undergoing ferroptosis, then released in the environment, and then somehow pass to a neighboring cell, bypass the threshold, and then activate RS generation again. So now, how do they activate RS again? You need some kind of nonlinear response in RS generation. And that usually comes from feedback loops. So then Hannah actually looked into literature to know what can be the feedback loops for the generation for RS. It involves at least one branch will be some kind of so-called signal in the kinases. Those kinases, it's an interesting fact in cells, is that if the cells need to grow, they need ROS, reactive oxygen species. We usually think it's really bad, but the truth is that if you don't have any reactive oxygen species, the cell will stop growing. You need some reactive oxygen. It's just like you need the kitchen to cook. But if the fire is too big, then you cook yourself, right? So that's like the system that the cells has evolved to actually control the ROS, right? Okay, so now that's literature. We want to know how those molecules are involved in controlling the propagation of waves. So we can add various different small molecules to actually perturb the wave. For example, in this case, we were talking about iron is important, so we decided to actually use iron chelators, DFO. After adding DFO, then you start to see the wave propagation become slower. And you can use the outline, you can see it's very obvious that initially it's very constant speed, but then after adding DFO, then gradually slow down and then propagate at much slower speed. We come up with all the response curve and showing that most of the time it's a dose response. But at high enough concentration of DFO, you actually totally stop the wave. You can also up back the irons, and then you can make the wave to actually travel faster, almost like twice as fast. And we also did various different small molecules to inhibit the so-called receptor tyrosine kinases. Those are the kinases that are usually, I was mentioning that they are important for cell growth. A lot of times cancer cells actually mutate those genes for the cell to grow faster. So we can have small molecules to inhibit them. We can also see it's mostly reducing the speed of the wave. But we want to know how can those kind of feedback loops that can lead to the emergence of a so-called bistable switch, right? So to this end, Jazo built a very simplified model where ROS generation is through positive feedback loops, and is controlled also by glutathione. And elastin is a small molecule that can inhibit the cysteine uptake, and as a result, drop the glutathione level. It's a simple partial differential equation model, reaction diffusion model. And the result is that we show it's possible if you actually starve cells, then you can start to have this switch-like behavior. So Hannah then go ahead to actually do the experiment to test this theoretical prediction where she used blue light again to introduce some ROS to the cell. And if there is a bistable switch, then cell would actually switch up, right? And this is the result. Before the photoinduction with increasing concentration of elastin, in other words, the stress of not having cysteine is gradually increasing, then you see the ROS steady state, it doesn't change before photoinduction. It's very, very similar. However, if you actually add a photoinduction, also you give them some external ROS that potentially can bypass the threshold, then you start to see this switch-like change at a higher concentration of elastin. So why is this important? And the theoretical prediction would be that with the bistability, you start to kind of have waves, and then you will propagate with a higher speed if the stress is stronger. However, if you don't have bistability, the death will be very local and will never spread. And this is actually consistent with experimental result. With a stronger stress, you have a faster propagation of the cell death with, again, with the lipid peroxidation wavefront. So for this part, I'll talk about, we developed a way that we can use blue light to initiate the cell death, ferroptosis. Yes, question? Yes, yes. Yes, yes, exactly. So that's my bad. Here, this is the Institute of Physics. I should show all the equations here. Actually, yeah. So what we know is that the nonlinear turn in terms of the Hill coefficient is very, very critical. The ultrasensitivity allows the multi-stability to occur. Without that, there is no possibility. Actually, that's one subject that we are actively studying because we believe the same thing about ferroptosis, but one chemical, elastin, that can induce the propagation of ferroptosis by engaging multiple positive feedback loop involving nonlinearity. But there's another way to induce ferroptosis without engaging a lot of feedback loops. In that case, the ferroptosis cannot propagate. You still have lipid peroxidation, but then it never propagates. It's almost like, so we are talking about elastin that can engage all these feedback loops, right? But if you only have, there is also a small molecule that can only induce lipid peroxidation. So it will engage only this feedback loop. In that case, you don't have propagation, and then cell death is a very graded response. We think it's very interesting behavior. And without, that's like super confusing for biologists. They are all ferroptosis, but one can propagate, another cannot. Why is that, right? But the truth is that you can induce the cell death in very different ways, but phenomenologically, they are the same. But the system behind is very different. And as a result, you have very different consequence in terms of, in this case, would be the propagation property. Yeah. Yes. Mm. Mm-hmm. Yes. 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 Or turbulent. Mm-hmm.
Yeah, we can categorize, but we don't depends on spatial state. First of all, can you categorize what kind of burning, low one, fast one, detonate, or terminal? It looks like at the end you terminal.
That's right.
Yeah, I don't.
Yes.
Answer from you.
Yeah.
Physics can have.
Yes.
Okay, for example, is the process exothermic, heat release, the process, the burning, or endothermic?
Yes.
Yeah, this may have
Very fundamental. Yeah, like, is there a change in temperature or energy release? How do we measure that? I think people. Yeah, that's very. Yes. Yeah. I think that would be super cool if we can measure those. As far as I know, I think there are Yes. To be honest, I think that is the big unanswered question that we really want to know better. Like, what really is contributing to this nonlinearity, and what can be the physical or chemical basis for this to occur? This is a fundamental question we don't know. And it requires very quantitative measurements, maybe the temperature, maybe the redox reaction. And we can use, from a biological science, we can use genetic or approaches or molecular approaches. But here we can use physical approaches to really know better about why do cells want to have like this kind of nonlinear bond type of behavior? We don't really know. Honestly, the challenge of the field is that there's no very good quantitative measurements. Even I show you those kind of pictures, we don't know exactly the concentration of RRS in the cell. How do we measure all those, and how does it change? We try to use better approaches that, so reversible reporters, we can measure steady state better and all that. But I think other perspective would be very useful for us to know better about this. And another thing I can tell people is that the whole field of Vertebrate, except us, are molecular biologists and chemical biologists. So when I go to the conference, I'm the only people presenting things that no one understands. But that's the development of the field, right? Because it's a very relatively young field, and we are the first group using systems approach trying to understand this phenomena. We are the first group presenting the Vertebrate can propagate in space, and then people were, honestly, why do we need to show 20 cell lines? Because one of the reviewer told me, I don't believe this. This is impossible. And we show in the first version 9 cell lines. So in the revision, we show 20. And including the cell lines, they say they never observed that. The truth is that they couldn't observe that because they didn't do a very fine time scale time-lapse imaging, so that they can not know the spatial pattern that actually can be observed, right? So I feel this is a field full of potentials. In my opinion, if we actually take a dynamic assistance approach. In fact, I think in biology, it's not only Vertebrate. I think there's so many things that if we can have a physics approach, it's going to make fundamental breakthroughs, in my opinion, right? Anyway, so I'm super excited to be here. Okay, so I was trying to summarize this. Basically, what I want to say is that we found there is a way we can use blue light to introduce exogenous ROS to bypass the threshold, so some of the cell will start to undergo ferroptosis. And through some kind of diffusing molecule that allows the bypass of the threshold in the neighboring cell. Once it bypass the neighboring cell, bypass the threshold, it will activate this ROS feedback loops. That's the nonlinear part of this. and then trigger the ferroptosis again, then it will release again RS-inducing molecule, and then so on and so forth. It will just propagate in space, just like domino effect. So I show you that this is we have some kind of priming signal, which is cystine starvation, and leads to the emergence of RS by stability, and that can lead to trigger waves, right? But I will use like a little bit 5 minutes to talk about the biological significance of this. I initially, after finishing all that, I was like, super cool, I love this result, I love everything, and then, but in our institute, in the Institute of Molecular Biology, my colleagues were like, that's all artificial systems, it doesn't make sense. Is it really happening in our body? That's the question they want to ask. I think that's a good thing that pushed me to really understand better, right? So if we really think ferroptosis can propagate as trigger waves without spatial boundary, then soon after the occurrence of ferroptosis, we will be all like this, right? So then that inspires us to go back to the literature that John Saunders once said, that during development of vertebrates, there can be elimination of tissue and organs that are temporarily useful, right? And in particular, he actually tried to describe this as a sculpturing of the limb followed closely upon waves of necrosis. At that time, people only know apoptosis and necrosis. If it is not apoptosis, everything else is necrosis. So people didn't know ferroptosis, necroptosis, all this, right? Pyroptosis. So we think maybe this is really what we see in the test tube, right? So we say, okay, let's just set up the chicken system in the lab. So we start to contact the agriculture department in our government, getting the fertilized eggs, and then start to culture the chicken embryo in the lab. It's actually quite fun. Then we can actually establish also ex vivo imaging. We use dental glues to actually glue the chicken limb so for us to image it. But we start by actually in vivo evidence. We look into the developing chicken limb using a reporter for ferroptosis called, it's a peroxide lipid 4-HNE. Use the antibody against this, we can detect the lipid peroxidation. It's actually happening in the middle of the chicken limb. It's pretty abundant. It's actually consistent with our ex vivo imaging when we fix we put a live limb out and under microscope, we start to see the cell death actually propagates. But it's very different from the cell culture situation. You know, the wave actually is constrained in the middle of the limb. We also show that if we add a small molecule that suppresses ferroptosis, the area actually go down, greatly diminished. And we did different controls to really make sure that ferroptosis inhibitor can actually really reduce the cell death in the chicken limb, but not apoptosis inhibitors. But why it is also limited in the center of the chicken limb? So one hypothesis we come up with is that maybe the fuel is only gathered in the center of the limb. Okay. So what can be the fuel? The oxidizable lipid, or so-called polyunsaturated fatty acid. So we measure the oxidizable lipid in the limb by using an assay called POP assay. Basically, you use laser to actually irradiate these areas, little dots, okay? And then you stain the limb first with a dye that when there is peroxidized lipid, it will light up. And this is what we see, and this is the quantification. You see the oxidizable lipid is actually centered around the center of the limb. So one possibility is that the organism evolved a strategy to actually have the peroxidized lipid centered around the limb so that they will only burn in the middle, and all the other parts are so-called wet trees, so they will never be burned. What? That's a big question. We don't know. We are right now trying to understand this. I think this is the mystery of life. Like, why is the fire only burning this way and only in the center? How do they form this? We have a hypothesis. We think, you know, in the complex organisms, a lot of time, pattern formation, morphogen signal, right? A lot of the time, those signals are forming gradients in space. they actually form a big area to specify specific cell fates. And that's what we think that can be potential mechanism. And what can be the function? Biologists want to ask what's the function, right? So we look into the occurrence of ferroptopsis. It's not only happening only in the middle of the limb. Actually, it's actually very abundant across different places. This is like a cross-section of the limb of different area. Maybe we can look into this one particularly. Those are the digits, those are bones. And this cyan color is the muscle cells. And those yellow are the ferroptopsis markers. You see, it's happening at the very surface area, the dorsal and ventral side, but it actually extends to overlap with the muscle. Okay. And actually, it's amazing that if you look into the chicken limb from day 6.57a, it grows super fast, and the muscles start to segregate. They are originally very continuous and start to segregate. And if you zoom in, you can see they start to segregate, and then you see those dots. Those little dots are the dead muscle cells. And if you do the overlapping between the dead muscle cells, those are the dots, with the peroxidized lipid marker, the ferroptopsis markers, you see more than 90% of overlap, suggesting those muscle deaths is actually caused by ferroptopsis. We want to stop this, so we actually inject ferroptopsis inhibitors into a chicken embryo. So how we do that? We can so-called candling. We look into where is the chicken embryo. We poke a hole and then start to inject. And this is a success. This is a failed one. You can inject into directly the so-called amniotic sac of the embryo. And after we suppress ferroptopsis, this is what we see. In the control, you see very beautiful muscle bundles forming like 5 bundles and very organized way during the development. However, if you suppress ferroptopsis, you have a lot more muscles. They are not dying, and at the same time, they are start to be a little bit crazy. They move around and not forming the proper shape. And this is what we see generally is happening. So that's the thing. So the field of ferroptopsis people, they all try to link to various different diseases, Alzheimer's, neurodegenerative diseases a lot, kidney failure a lot also. Muscle, I don't know if muscle is actually people study yet. I think probably our study is the first one, but this is in a so-called physiological context, right, during development. And actually, our study is the first physiological context that people observe. Right. So what we think is that maybe what happens when we want to build some complex and big embryo individual, you actually, just like building a bridge, right? So you need some temporary structure that is big, and so that you can lay the groundwork for building a bridge like this. Once the bridge is actually solid and stable, then you can remove the big ones, the big temporary structure, and those can be through the mechanism of ferroptopsis trigger waves. So here I'm showing you maybe the priming signal in the real physiological context can be morphogenetic developmental signal that leads to redox by stability and leads to the propagation of the wave in a very spatially regulated way. So we are very interested in knowing what can be the biological regulation of this. How this biological system evolve various different control mechanisms to actually confine this kind of large-scale cell death in a specific region and in a very precise way. I think this is amazing to know. And we want to know more quantitatively the nonlinearity of this RS generation. Probably chemically and physically, I think that would be super cool. And finally, we want to be able to control this. If we can have some kind of circuits or some kind of engineered cells, we can control this, it's going to be amazing. The reason being that we can control it to occur in the case of, say, cancer cells. The cancer therapy a lot of times is because of fractional killing. Some of the cell never responds to chemotherapy drugs genetically or non-genetically. So if we can induce this wave, it will actually eliminate a lot of cells at the same time. But we can also prevent large-scale cell deaths. And what we think is that a lot of times in the so-called degenerative aging-related diseases, it can cause large-scale cell deaths probably through also this kind of redox by stable switches. If we can prevent this, then it will probably also be very helpful. We have some kind of preliminary data regarding to the question initially people raised in the back about density and alignment. I was mentioning we are very into this kind of self-organization of cellular behaviors. And this is one kind of birds that every time they will gather on the sky of Rome in Italy, and then form this kind of beautiful patterns on the sky, right? And the truth is that cells can also do that. If you actually monitor the cells this is 2 to 3 dimensional cell growth, you see cells are forming patterns that are trying to escape from the center, or they are forming this kind of tornado-like structure, right? It's amazing that cells can behave like this. But under this condition, when we introduce Vertebrate cell death, this is what we see. The cell death actually will turn the directions and change the speed and forming patterns. To me, this is super interesting in a way that cellular behavior actually drive the occurrence of cell death in a very, I would say, very regulated, unexpected way. So we are actually currently trying to understand this in a very detailed manner, if possible, that what can be the mechanism drive the initiation of the cell death, as well as how do they propagate in space in a way that can be modulated. And finally, we are interested in knowing what's the nature control of this cell death. So, I mean, in biology, the textbook knowledge of so-called interdigit regression, then usually our hands are like, like Doraemon, right? But then over time during development, the cell would die during the digits, and then you start to separate. And it has always been said it's apoptosis. People always say it's apoptosis. The truth is that when we actually suppress Vertebrate, now the chicken now become Doraemon, right? So we are not saying, I don't think it's only Vertebrate. It's actually multiple cell deaths may occur at the same time in a hierarchical or simultaneous way to actually regulate the interdigit regression. And we need to use multiple tools to understand this better.
So, with animal, you give a
yes. Yes. So we do know some part of we have some understanding of this. Like, for example, duck and the chicken, they are very different, right? And people did some study or the frogs, right? So people understand that oxygen level is very critical for the interdigit regression. So oxidative stress is actually very important, right? But then oxidative stress can induce both apoptosis and ferroptosis. So how does this two occur in a coordinated way to mediate this regression is something we don't know, right? The truth is that we actually try to understand this in the very beginning instead of understanding the muscle regression, all that. But it's very difficult to study this because we cannot study this ex vivo. They don't form in the ex vivo context. And it's hard to image. It's a 3-dimensional space. It's pretty thick. It's hard to actually image it as a dynamic process. So it becomes difficult. You need to use other approaches to study it. But I think that is something we, of course, are very interested in knowing better. What? Time scale. Yeah, so in vitro and in vivo, it's all about several micrometer per minute. Several micrometer. A minute. A minute. So it's actually slow. It's a slow wave. Okay, finally, I would like to thank people in my lab, Hannah and Jiazhou, who are, as I mentioned in the beginning, they are really the heroes behind the scene. And in addition to Vertebrate, we are also interested in a bunch of different things that, like oscillators I was talking about, or long-term time series analysis to understand the information encoded in signaling dynamics, those kind of things. So with that, I would like to take any more questions you may have. Thank you.
Thanks, Sheng-hong, for very inspiring talk. Okay, we will take a few questions.
Okay, thank you for your talk. And here, I'm wondering, for the trigger wave, is it only happen within the same cell types, or is it possible for
very good.
Yeah,
Yeah,
between
good
different
question.
cell types?
Yeah,
Thank you.
right. So a researcher student of our lab actually did the experiment. So we could see it occurring across many different human cells. And if he actually put A cell and B cell, start a wave in A cell, it can actually propagate to B cell, induce the wave. So suggesting probably the molecule that they secrete out is something general across different cell types. Yeah. They do have different thresholds. We are measuring that. We are measuring different, the cool thing about this, another thing is, the threshold is dynamic. So we think it's a so-called dynamic bistable system. So the curve of this bistability, it will change, evolve with time. Yeah, so in other words, the RS system keeps changing over time, and then you change the threshold. Yeah. Oh, you mean the last part? Yeah. That's true too. Yeah. That is true. Yes. Exactly. 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 Yes. Mm. We don't know. Yes. Right.
Yes, yeah, an interesting talk. Yeah,
Thank you.
Physics audience, the physicists can associate this phenomena with quite a few physics phenomena, just like Kevin suggests. In fact, laser, you can see that in a broad sense, also work like this. But the question is do we know how universal it is? It means that the key message seems to be that massive cell death is due to trigger wave, due to a bistable system.
Yes.
And in your case, that is induced by the lack of glucose. So this is the chain of events. And then you study it well and the details. So do we know how generic, how universal it is?
Yes.
For other cell death or other things that has nothing to do with glucose and so on?
Exactly.
Do they behave like this?
Exactly. That's a wonderful, wonderful question. I can see that as a great physicist asking a question. What? Oh,
Yes,
high energy.
Right. To answer,
Right.
every photon in the universe behaves the same. So I like
Okay.
talking like this.
Okay. Yes. We are right on that. So, of course, we want to show across different cell types it occurs. But this is a chemical reaction, chemical-induced bistability. We want to generally examine all different possible ways for the RS to have bistability. What are the different ways that we can induce RS bistability? That's one really burning question we are asking right now. And we actually have some results showing that there are multiple different ways that you can induce the bomb. But then the question is, right now we are using chemical ways, like small molecules suppress certain metabolic processes, all that. But the biological system must evolve some way to induce or control those processes, right? I feel those processes will be a lot more robust for us to control it, right? So we want to find that, right? But we are using chemical, genetic, all different ways to try to know
And
how general we can
induce.
induce.
It's
What?
trigger wave. its trigger wave would subsequently kill the cell. So
Yes. So when we see it, we actually, our output is not by stability. Our output is propagation of cell death, and to start with. And then we go back and know that is oftentimes is bistable. So in other words, these two, at least in our hands right now, occur at the same time. Bistability and propagation happen at the same time.
So yes, right.
Yeah.
This is a good direction too.
Thank you.
Yeah. Show universality.
Yes. Yes. We want to know how general this is for sure. Yes.
Any other questions? Maybe I can ask one question. So,
Fantastic.
I think one of the main purpose of this ferroptosis, as you mentioned in the beginning, is to remove some of the intermittent organs or tissues during the development. So you also tested 20 different cell lines to show the generality of.
Yes.
But because all these systems are in the mammalian cells, so
they are all human cells.
Oh, human cells.
They are all human cells.
So my point is it possible?
What? Oh, the tissue culture are all human cells.
But this phenomena could this happen in the bacteria, like biofilms?
Yeah, I think that would be super interesting. Fenton reaction is chemical reaction. It happens everywhere.
Yeah.
Right. So and that's another really interesting aspect of this is that the redox reaction is evolutionary so conserved. It happens so early on, and iron metabolism especially. It's amazing that iron detection is so conserved from bacteria to human. So I think this is something probably very fundamental to all forms of lives. I don't have an answer for you, but one thing we want to really know right now is we want to see this ferroptosis from human to, this summer, our summer intern is going to work on this. From human to mouse to zebrafish to Drosophila to different organisms. We want to see how this can be a general strategy during development that can be eliminating important temporary cells. Yeah. Bacteria, I don't know. Maybe biofilm will be very interesting because they are really interesting patterns.
That's right.
Yeah.
A nice talk and very attractive pictures.
Thank you.
So I have a question, maybe not related directly to this talk. So whether it is possible to control the killing of some specific type of cell, so this can be used to treat the cancers.
Yes, that's what we are after, of course. We want to know, it's not trivial, but we really want to be able to target cancer cells and then induce these large-scale cell deaths. Right now, we are mostly thinking about, as I just briefly mentioned, about how we can have a circuits. So there are several ways we can do. Okay, so first thing we are doing right now is that we actually go through a bunch of different cancer cell lines. Some of them are more sensitive, some of them are less sensitive to ferroptosis. We want to know what can be the molecular fingerprints for the difference in the sensitivity. And once we know those, and can we harness the differences and generate some circuits and go into cells so that it can make them universally sensitive. That's what we are going after right now.
Thank you.
Yes.
Thank you very much for a great talk.
Thank you.
I might miss this during the talk. So I noticed that for your trigger waves, you can see the different density gradient, and there's some width for front. So can you say more how those front width and the density of fronts or speed of fronts depend on the parameters like a nutrient, the pathway parameters, rates, and so on? Can you control these quantities?
And so I'm trying to interpret your experiment. Your question. Your question is, how does the wave? I show the anisotropic propagation of waves in a self-organized cell population. So the question is, how does density and cell alignment affects the propagation of wave and also initiation? How to quantify?
The wavefront, the detail of your wavefront.
Yeah. Right. How do we quantify? It's a lot of image analysis. Is that what you are asking? Like, we did a lot of image analysis.
Like you also do simulations. So can you know what are the important control parameters
Yes.
control
Very good.
all these quantities?
Exactly. Right. So we are wrapping up this study. So the anisotropic propagation, at least we are just go back to simple things. First thing is density. Second is alignment, cell alignment. You can see very nice cell alignments, right? So we did correlation study first and to see how the density affects the propagation. So the truth is that when the density is high, the wave will actually stop. The cells at high density, they are more resistant to ferroptosis. And there is some molecular basis for this that people already know, okay? But another thing we observe is that if the cell align like this, the wave will propagate like this. It will propagate along the alignment. That's our analysis, and we have a hypothesis for that, which is that, what? Polarization of the cell, and the polarization of the cell leads to a polarized distribution of lipids. And the lipids at the polar ends are usually the oxidizable lipids. So you can imagine once the RS comes in, then it will burn the polarized ends. And it's almost like, if you look at the cell, it's actually anisotropic diffusion of the RS signal because it's polarized. And another really cool observation we have is that if you look into this spindle shape of the cell, they are really like spindle shape, and then this periphery, that's the flat sides, is actually very rich in the saturated lipids. Those are the lipids that cannot be burned. It's almost like there is a firewall. So you have a firewall at the flat side of the cell while you burn in the middle, at the polar ends. And we did some agent-based simulation, and we could see if you have this kind of heterogeneous or polarized distribution of lipids, you actually can drive the propagation of the cell death following the cell alignment. And we are based on some observation, and then we did some simulation to show it that way. That's what we think right now. Yeah.
So following the questions, I can think about two possible mechanisms for the uneven lipid. One is the saturated lipid. It's more on the side.
Yes.
It could be because you want to enhance the hydrophobic interaction. Another possibility is the charge, because we do know some lipid carry a charge. And distribution of a lipid will depend on geometry, because they mutually repel each other. So the apex region will be the lipid carry more charge. And that one could be vulnerable to oxidation because they carry charge.
So
I don't know. So
how is charge linked to the saturation of the lipid?
So charge doesn't link to saturation. It's just, like for example, PIP2, PIP3, usually are saturated.
Right.
But are easy to carry negative charge on the surface. But because the negative charge tend to distribute at apex, the negative curvature region. Because
Right.
That's
Right.
two possibilities.
Right. I see. Interesting. We'll think about it. Yeah, sure.
Okay, I think due to the time limit, we'll stop here, and let's please join me to thank Sheng-hong again. Okay.
Thank you.
And we have also refreshment outside. We can continue the conversation or discussion outside in the lobby. Okay, thanks.